All right, well, good afternoon, everyone, and welcome to the HNRCA Monday Seminar Series. Today, it is my pleasure to introduce Dr. John Cryan as today's speaker. Dr. Cryan is Professor and Chair, Department of Anatomy and Neuroscience at the University College Cork, and has been Vice President for Research and Innovation since 2021. He is also a PI at the APC Microbiome Institute in Ireland. He received his bachelor's in honors in biochemistry and PhD in pharmacology from the University of Galway, Ireland, and was a visiting fellow at the Department of Psychiatry in the University of Melbourne, Australia. Following this, he did a postdoctoral fellowship at the University of Pennsylvania and Scripps Research Institute in La Jolla. He spent 4 years as a group leader in the pharmaceutical industry with Novartis in Basel, Switzerland, prior to joining UCC in 2005. His current research program has been a pioneering force in the field, which focuses on understanding the interaction between brain, gut, and microbiome and how it applies to stress, psychiatric, and immune-related disorders at key time windows across the lifespan. Dr. Cryan has published over 700 peer-reviewed articles, co-edited 4 books, and is co-author of the best-selling The Psychobiotic Revolution: Mood, Food, and the New Science of the Gut-Brain Connection. He's an elected member of the Royal Irish Academy in 2017 and is past president of the European Behavioral Pharmacology Society. In addition, he has received numerous awards, including the Royal Academy of Medicine in Ireland and FASIB, and he has been on the Clarivet highly cited research scientist each year since 2017. Furthermore, he's a TEDx speaker in Washington in 2014 and was profiled in the Netflix documentary Hack Your Health: The Secrets of the Gut in 2024. And today his talk is titled Gut Feelings: The Microbiome as a Key Regulator of Brain and Behavior Across the Lifespan. And before we jump into the seminar, for the audience, please place your questions in the Q&A box and we will respond to them at the end of the seminar. And so without further ado, welcome Dr. Cryan, and we look forward to your seminar.
Thank you, Jen, and thank you to all your colleagues at Tufts for this really nice invitation today. And with daylight savings, we're only 4 hours difference from Boston, so that's a nice thing overall. So yeah, I'm based in Cork in the south of Ireland, and I'm going to just take you on the journey that we've been on here over the past 2 decades, implicating what's going on in the brain, what's going on in the gut, with what's going on in the brain overall. And I like to start with this story. It's a story of the painting Le Bateau by Matisse, and it hung like this as part of a retrospective exhibition in 1961 at the Museum of Modern Art in New York. And many people came to see it, including Matisse's son. And it wasn't until a very eagle-eyed stockbroker called Genevieve Habert came by and noticed that something was wrong. And what was wrong was that the painting was hanging upside down, and people have been looking at it the complete wrong way around. And so my first take-home message to you today is really that to remind you that sometimes we have to turn things the other way around to see how they really work. And that's kind of how I frame our work on brain and behavior overall. And let me not do that. Okay, and I work like many of you. I work in a medical school, and in medicine, everything goes back to Hippocrates, who's taught to have said, no, it could be fake news, but he's taught to have said that all diseases begin in the gut. Now, when I talk to my neurology or my psychiatry colleagues, they would often question that. And my neuroscience colleagues, who are by far the most skeptical of all, often challenge me by saying, John, if you're so interested in the neurobiology of emotions, why are you so fixated on the gut? But all we have to do is look to our language. We use phrases like gut feelings and gut instincts. We're gutted when we're disappointed. We make gutsy moves when we're brave. We have butterflies in our tummy when we're nervous. So perhaps there is an underlying biology to all of this. And in the last 2 decades, we've seen a quiet revolution occur in all aspects of biomedicine, which has been the realization of the importance of the microbiome in all aspects of health and disease. And with the microbiome, the thing I want to remind people is that we're living in a microbial world. microbes were here long before humans. And so, if I share with you some of the statistics on microbes, they are really quite humbling. If you looked at all the genes we have in and on our bodies, we would be more than 99% microbial. We have more microbial cells than we do human cells. So it is really an important aspect to take into account. And some years ago, I had the pleasure of being on this framework document, really, which tries to contextualize from an evolutionary perspective how we as humans, with our host genome, coupled with our microbial genome, make up a hologenome or a holobiont that has to navigate through an environmental metagenome. And again, once you start conceptualizing things within this framework, you can really start to operationalize things a little bit different. And what my lab is interested in is how the microbiome is affecting brain and behavior at key windows and transitions across the lifespan. We have programmed in the perinatal period, in adolescence, in adulthood, and all the way to aging, which I'll try and focus most of the talk on overall. For the most part, we're thought to be sterile in utero, and we get our microbes as a kind of a birthday present from our mom on the day we are born. And then that microbiome takes some years to become regularized and stabilized, and then follows us through our adulthood. And then as we age, again, we start to see how that microbiome starts to lose its diversity. And we're beginning to try and parse this relationship between what's going on in the microbiome with key aspects of brain function across the lifespan as well. And some years ago, we began to focus the aging brain in the context of stress. I'm a stress neurobiologist, and one of the things we started to realize that where stress and aging converge could be really important in terms of the behavioral sequelae that occur, whether it's in cognitive impairment, anxiety, depression, or it's the social brain in particular. And one of the places they start to converge is at the level of the gut-brain axis, and in particular in relation to changes in the microbiome. And that was really kind of an interesting point for us to start thinking about what is going on and how they actually overlap. Moreover, we're beginning to understand all of the factors that can influence the composition of the microbiome across the lifespan. Many of these which are environmental, as well as different exposures, the effects of medication, infection, climate, activity, and particularly exercise, circadian rhythms we'll come back to. I already mentioned stress, and of course diet, which is of relevance to this audience as well. And diet is one of the key factors that influences microbial composition, and then how that can affect how it communicates to the brain. Now, going back to my skeptical neuroscientist, so we have to come up with stronger evidence that the microbiome could be important for brain function and brain development and brain aging. So how do we get more evidence that something is involved in a particular process? Well, if we look to engineering or other areas of biology, what people do is they take it out and see what happens. And so the concept of growing up without microbes, growing up germ-free, isn't new at all. It goes back to Louis Pasteur's time. Here you can see a germ-free man from a sci-fi magazine from 100 years ago. Now, he doesn't look that great. And while our germ-free mice, which we work on, have their own problems, they do look a lot better than he does. But the one thing I would say about using germ-free animals is that there's really no human translation, with the exception being the boy in the bubble that Paul Simon sang about in the 1980s. But these mice in the bubble have been a really important part to answer the question, is the microbiome involved? Yes or no? And have taken me and my lab to all kinds of really weird and wonderful places that I would never have expected overall. Places like where? Places like the wonderful world of myelination. We showed over a decade ago that if you grow up germ-free, there is an increased myelination occurring in the prefrontal cortex that seems to be very prevalent in the male brain. More recently, in the last months, we've shown that this also occurs in germ-free zebrafish, and I've looked at the developmental sequelae of this. It's in a recent paper, and this is this is key because we mainly think of myelination in the context of demyelinating disorders like multiple sclerosis, but here we see this hypermyelination occurring. Adult hippocampal neurogenesis, the birth of new neurons that occurs throughout the lifespan. And we know as we age, in particular, we start to see a diminution of the neurogenic potential in the hippocampus. And we showed some years ago, Olivia O'Leary, who's on faculty here now, showed that there is a dysregulation of the neurogenic mechanisms in the hippocampus, which may be relevant to cognitive processes like pattern separation in particular. A variety of synaptic plasticity related proteins and neurotransmitters, many of which are involved in learning and memory, are gone awry, whether it's GABA or serotonin, or we get a loss of brain derived neurotrophic factor in these germ free animals, changes in dendritic arborization and dendritic growth, microglia, which are really important part of the brain's immune system. They're quite immature and are not functioning appropriately without these microbial signals. And as we showed here in this review article in Nature Reviews Immunology last year, this relationship between the microbiome and microglia could be very important in synaptic pruning aspects overall. And then finally, blood-brain barrier, which is also really important for maintaining healthy brain aging. That it's been shown that the permeability of the blood-brain barrier requires the appropriate permeability requires having microbes in the gut. I used to say that my lab, we're not known for our focus really, but we did everything except electrophysiology because I felt there was enough electrophysiologists in the world. But anyway, I was waiting for the electrophysiologist to jump on this really exciting area of microbiome brain interactions, and I waited, and I waited, and I waited, and so a bit like Godot, they really never came. And so luckily I recruited an electrophysiologist into my team at the time, Ken O'Riordan and Henry Darch, and we were able to look at slice electrophysiology again in the hippocampus because of its important role in learning and memory. And what we found was quite a remarkable finding that the microbiome gates a normal long-term potentiation, LTP being a cellular correlate of learning and memory. And if you're a male mouse, in dark blue here, you can see the a normal response, LTP response, but in light blue, in a germ-free animal, they're not able to make this response. If you're female, you can. So that's quite interesting. And that raises a whole other aspect of the microbiome brain interaction, which I'm going to put my hands up right now and say that we just don't understand at all. But it's really intriguing that there's something so different between the sexes in relation to how microbial perturbations are influencing brain. And this is something that deserves a lot more attention. And there's something about the male brain that makes it quite vulnerable, particularly in early life, or the female brain that's quite protective. And we know that the microbiome is really important for sex steroid metabolism, and we have an estrabolome, and this may be playing a role. But also, it opens up questions around menopause, around cognitive function, and other as we age, and in age-related disorders that are more prevalent in women, such as Alzheimer's disease in particular. So this is something that deserves a lot more attention. Okay, here's just, you don't need to be a neuroscientist to see this very clearly. This is a basolateral amygdala neuron, and the amygdala is the region of the brain involved in social behavior and fear learning. And you can see already that it's an arborized state if it's germ-free. It's not functioning appropriately. It's almost like it's in a stress state already. And then we were interested in trying to parse what that would mean at a behavioral level. And it's not surprising then that what it, in our mouse models, we can see that fear learning deficits occur in these animals. The animals could learn okay, but they couldn't actually recall appropriately or extinguish the fear memory. And in relation to social behavior, mice, like humans, are quite social. So if you give them the chance to spend time with another mouse or an empty chamber, they'll gravitate towards the other mouse. But again, not if they're germ free. And mice, like maybe some humans, are a bit fickle. So if you give them the chance to spend time with their everyday playmate versus a new playmate, they'll gravitate towards the new playmate. But again, not if they're germ-free. And so this, for us, was really important animal studies to show that for normal social interactions and you're a mouse, you need to have appropriate microbes in your gut. And that led us to start collaborating with zoology and microbiology, evolutionary microbiology colleagues, and in this quite speculative paper at the time in Science, we were able to survey across the animal kingdom. And if you, whether it's a bumblebee in a hive or a baboon in the wild in Africa, if you start to interfere with social dynamics and social hierarchies and social structures, that you start to change the composition of the microbiome for sure. And vice versa. If we start to tinker with the microbiome by various mechanisms, we find that the changes seem to be very related to the social brain. And there's something about signals coming from the microbiome to the social brain that are really important. And while today you're all on Zoom, it would be quite different if I was live there in Tufts. We would be and we were in a crowded room, you would all be spreading your own aura of microbes from one to the other. But today you're spared that, and just think then you can't be sharing these social microbes from one to the other and what the impact that might have overall. And we've been very interested in disorders of the social brain. Of course, this is most talked about in the context of autism spectrum disorders and neurodevelopmental disorders, as well as adolescent onset schizophrenia. But one disorder that's not talked about a lot is social anxiety disorder. It's a very common disorder. It affects about 12% of the population. The biology of it is not unclear. And social anxiety disorder is a disorder where people have an intense feeling of being judged in public. It's not shyness, it's not introversion, it's just a feeling of being judged by others. And it's very disabling. And we were interested, would the microbiome play a role? And we showed, this is work Mary Butler, who's a psychiatrist now back here in Cork, showed that if you looked at the composition of the microbiome in patients, well-phenotyped patients with social anxiety disorder, there is a difference overall. And that was kind of intriguing and interesting in itself, but it's just a correlation. And so the next thing was to see, could we test this? Could we actually parse this and get more causal in our studies? And if the microbiome changes in social anxiety disorder are relevant to brain and behavior, surely we should be able to take the microbes from these patients and give them to normal mice and see maybe it could change anxiety or social behavior. And Nate Ritz, who is from New Mexico and is now postdocing in Seattle for his graduate work, took on this challenge. And what we found was that in an animal model of social fear extinction developed by David Slattery's group, again, a former postdoc who's now in Frankfurt, we were able to show that in animals that got the microbes from patients with social anxiety disorder, they have a clear deficit in social fear extinction, which was coupled with changes in oxytocin and vasopressin in the brain. Really remarkable, really specific to this type of behavior, and helps us build a causal inference that the social brain in particular is sensitive to these signals. And so we began to kind of triangulate that the microbiome is important for brain development and function. Microbiome is involved in social behavior. We know from Robin Dunbar's theories from Oxford that the social brain is really important for cortical development. And so we can start to triangulate all of this. And that leads us to really provocative questions about, can we understand the social brain more by these environmental and other factors? And intriguingly, can we influence the social brain through the microbiome overall? And this is where diet and nutrition are going to play a key role, because we know the social brain starts to deteriorate as we age. And one of the big problems of aging is in a deterioration of social structures due to loss of social partners, and various social interactions become less. And so the question is then, can we influence the microbiome to promote the social brain to become more social, and so to allow for more empathetic or more cooperation or more trust? Or could we ever get the Irish football team to win again? And all of these are questions that we ask about this. And maybe that the real social network isn't Meta or Facebook, but is actually the microbiome overall. So that's a lot of our work on this, and we have some good data coming from early life that diet is a way in here. And we've shown, for example, if you give antibiotics in early life, it causes impaired social memory and a reduction in genes involved in learning and memory. And then one of the things we've shown is that if we give a diet, and this time it's a diet which involves prebiotics, which stimulate beneficial bacteria. In this case, it's called GOS and FOS, as well as sugars that are really important and present in breast milk. That it can, when we do that, it can enhance social recognition memory and genes involved in memory overall. And this could help develop, counteract the effects of microbial disturbances in early life and more or less create a more resilient microbiome. Similar work in this collaborative work with my colleague Harry Shellikens, we were able to show that the negative effects of a high fat, high sugar diet in early life on the microbiome could be countered by giving specific strains of bacteria, in this case a bifidobacterium longum, and prebiotics, in this case GOS and FOS as well. This was a paper just out in the last week or so, really showing how we can help through nutrition get a more resilient microbiome to influence the social brain. Because of this audience, I'm going to fast forward to the extreme of life of aging, and of course this goes to the work of Eli Metchnikoff, which many of you will be aware of. But if you're not, don't worry. He's the father of modern immunology, and he was he grew up in what's now part of Ukraine. He went to the University of Kharkiv, but spent most of his working life in Paris, working at the Pasteur Institute. And he won the Nobel Prize for showing how immune cells gobble up foreign bodies and this process of phagocytosis. And in doing so, Metchnikoff became one of the great fathers of modern immunology, although he was originally trained as a zoologist. Now, when he turned 50, he got very worried about getting old. And he was like, look, as an experimentalist, was kind of trying to figure out, there must be ways of trying to figure out, are there parts of the world that age better and healthier? And he went across Europe and he wrote about it in this book. And one thing he noticed was that there are parts of Bulgaria, in particular, in these villages where people seem to live longer. And what he put it down to was what they ate. And what they ate was a lot of fermented foods containing lactic acid bacteria. So he's a father of not only immunology, but author of kind of aging science or geriatric science and health science in some ways, because he was using methodologies to actually chart a natural history of aging. And also the father of probiotics. And if you go to Korea, you can get a yogurt drink with his picture on it, which must have that Nobel sign of approval and must be good for you overall. But the thing with Metchnikoff, like many great scientists, he was full of many crazy ideas, and this was just one of them. And so most of this got forgotten about for at least 70 or 80 years. And it isn't really until the last 2 decades that we began to realize how important the microbiome is for healthy aging across all systems in the body, whether it's the immune system, the cardiovascular system, the gut, or the brain. And there are many studies in this, and I just highlight one, which is from my colleagues here in Cork, led up by Paul O'Toole. Some years ago in Nature, they did the ElderMet study where they showed that individuals who lived in predominantly a nursing home environment, that the microbiome was less diverse. Now, I know diversity is somewhat of a loaded term nowadays, and it shouldn't be, but diversity is really important in all aspects of our lives, but in particular in relation to our microbiome. And what Paul and colleagues showed was that the more diverse the microbiome, the better the health outcomes, especially on indices of frailty overall. So then that leads them to look at it in another way and see what was driving the diversity of the microbiome. And then what they put it down to was the diversity of the diet. More diverse the diet, more diverse the microbiome, and the better the health outcome. And this wasn't a good public health statement for people in the nursing homes who eat a lot of beige food, and not a very great diversity. So this is a real important finding overall. So we, as an experimentalist, we were interested in revisiting Metchnikoff in the context of the work from Paul and colleagues, and we are also able to show that we could correlate the age-related changes in brain behavior, inflammation, gut barrier function, etc., with the microbiome in mice. And that was pretty cool. But it's only a correlation. So the next step was to, can we intervene? And so what we did was we looked at inulin. Inulin is a prebiotic that stimulates beneficial bacteria, and it's found in leeks and artichokes and chicory and many vegetables. And we showed, and for this study, I turned to middle age because for obvious reasons, it's a time window that I've been interested in of late. And what we already found in the middle-aged brain, that it's already starting to show signs of neuroinflammation. There's already an infiltration of inflammatory monocytes and an increased microglia activation. And what we found was when we treated these middle-aged mice, these are 11 to 13-month-old mice, with inulin, we could dampen down this neuroinflammation overall. And this is work again led out by Marcus Boehm, a postdoc in the lab at the time, with my clinical colleague here, Ted Dinan, and Harriet Shellikens, a close collaborator as well. And really the take-home message here is that if you're thinking of having a midlife crisis, maybe forget about the motorbike and turn to the leaks instead, and it might be better for your brain overall. Now, I was very buoyed by these findings at the time because they help build our argument towards a more causal relationship between the microbiome, diet microbiome, and healthy brain aging. But we also know that diet can affect the brain directly, and not everything has to go through the microbiome. So how can we prove that it's the microbiome? And for this, then, Marcus here, who's here on the right, I convinced him to do this experiment where I told him, you're going to spend a year of your life doing this experiment, and it's not going to work, but we have to do it just to prove one way or the other. And that experiment was that if the microbiome holds the secret to healthy brain aging, then we should be able to take the microbiome from a young mouse and give it to an aged recipient and be able to rejuvenate the brain in some way and shift behavior. And much to my surprise, pleasant surprise, that's exactly what happened. Here we went out to 22-month-old mice, and we took the poop from mice that were 8 weeks old, and we were able to, by transplanting that in, we were able to shift the aged microbiome, make it more compliant, and then we were able to reverse or change the peripheral immune system, the central immune system in terms of microglia function. But most surprisingly, when we looked at the metabolome in the hippocampus, the region of the brain involved in learning and memory, we completely rejuvenated that hippocampus, as well as having positive effects on social anxiety and cognitive behaviors. Now, don't get me wrong, not everything was amenable to being reversed. There's lots of supplemental material in this paper of things that didn't become reversed, whether it's neurogenesis, adult hippocampal neurogenesis, or telomere length, or various other things that we thought would. But really remarkable, we were able to show this. And 3 other groups subsequently have shown this, including groups in Freiburg, at the Quadrum in the UK, and in China. So this is really, really intriguing overall. And then more recently, my colleague Yvonne Nolan here and I was involved somewhat peripherally in this study, did the opposite, where they took the microbiome from patients with Alzheimer's disease and gave it to normal, healthy rats in this case. And what they did was they were able to bring on hippocampal dependent cognitive dysfunction in these animals when they got the microbiome from Alzheimer's patients compared to when they got it from healthy controls. So again, adding this evidence to what we're doing. Now, before you all rush out and start to steal the poop from your graduate students or your grandchildren or whatever. That's not what we're saying here. This is a proof of concept that the microbiome is really important for healthy brain aging and that we need to mind our microbes to look after our mind as we're aging. Some of the other aspects we've been building into this has been looking at how nutrition could be playing a role. So again, looking at these prebiotic approaches. So again, we used phosphinulin here as an approach that to see that could we attenuate the effects of stress in aging on brain and behavior, and in particular social behavior. And with stress induces these deficits in aged animals. And indeed, we could rescue this through giving a prebiotic diet. Again, telling us that the microbiome is amenable to protecting the aging brain, at least if you're a mouse. And Joanna Pereira, a graduate student at the time, also was interested mechanistically in this. And this is an intriguing finding, which we showed at the time, which is kind of gave us the opposite results of what we expected, because what we found was that when we obliterated the microbiome in aged animals, it was not having a negative effect, but having a positive effect. So aged animals have deficits in their social behavior, and when we got rid of the microbes, we were able to rescue this. So we're not recommending giving broad spectrum antibiotics for the aging brain, but here this proves and to get at some of the mechanism, we also showed changes at the level of infiltration of immune cells across the choroid plexus in particular, and having a neuroimmune basis to this. Really intriguing data, and it helps prove the concept that there's probably something not right in the aging microbiome, and we might need to target that to help healthy brain aging. Now, here in Ireland, we don't have the Metchnikoff and other Nobel Prize winners for physiology and medicine, too many of them anyway. But we do love to talk about our Nobel Prize winners in literature. And William Butler Yeats won the Nobel Prize for over 100 years ago now. And Yeats was a poet who was very fascinated by complexity. And he really like to deconvolute the different aspects of complexity, whether it's political complexity or in this case, the complexity of nature overall. And here he was trying to, in this poem, he's trying to deconvolute whether a chestnut tree is the leaf, the blossom, or the bowl, or how can we really know the dancer from the dance. And I like this because it reminds me that what we're trying to do in getting at mechanism in how the microbiome is signaling to the brain, it's really about deconvoluting this complexity. And I was able to sneak this quote into a neuron review some years ago, just to highlight how difficult it is to look at the relative contribution of the various pathways of communication between the microbiome and the brain. And that's something that we're really interested in my lab. The two big things my lab are focused on right now are trying to understand these mechanisms and then trying to move towards human translation of our really exciting animal studies, and we're beginning to scratch the surface. But really, it is a very exciting time in microbiome brain effects, especially in the context of aging, I will say. And there's a lot of exciting work emerging and will emerging, not just from our own group, but from many other groups across the globe right now, really building concrete evidence about how microbes can send signals to the brain to either help cognitive processes or hinder cognitive processes at key transitions across the lifespan. Now, one of the ways of communication between the gut and the brain is through the vagus nerve. Now, vagus comes from the Latin for wandering. It's a long wandering nerve, same origins as words like vagrant and vagabond. And we showed now 15 years ago the importance that the vagus nerve has in gut-brain signaling. When we showed that when we cut the vagus nerve, all of the effects of a specific bacteria, in this case a lactobacillus, were gone. And this reminds us that what happens in Vegas doesn't necessarily stay in Vegas, and that it will affect our emotions in key ways, and that this is a key pathway of communicating between what's going on in the gut and the brain. What we need to really understand more is what happens when it gets to the brain, and that we've ongoing work trying to deconvolute what's going on at the level of the nucleus tractus solitarius, and how does that impinge on cognitive and emotional circuits in limbic areas. And then moreover, how do you get to the vagus, and what sends the signals from the bacteria to the vagus, and how that occurs. And one of the ways that that can occur, and in general, is through the metabolites that the microbes make. And it's important to realize that your microbiome is like a little factory producing all kinds of weird and wonderful chemicals that your body wouldn't be able to make otherwise. These include short chain fatty acids, for example. And so we've been able to use this as an approach to see, well, what if we bypass, because I'm a pharmacologist by training, so I'm like, what if we bypass the microbes altogether and just give animals these short chain fatty acids? And indeed, we were able to have positive effects, and people are now testing these, and there are many other metabolites. And then how do these metabolites activate the vagus to activate the brain, and really become a conduit for body-brain interactions and key interoceptive signaling that is really important for body-brain interactions. Another thing worth mentioning is in the context of barriers. Now, Maria Aborto came to our lab here in Cork as a postdoc, and Maria is fascinated by two things, or many things, but two of her main things are barriers and Lord of the Rings. And so in this review, which she takes us through the various gates of interaction, the barriers, whether it's gut barrier, choroid plexus barrier, or blood-brain barrier, and shows how the microbiome could be really playing a key role in shaping the integrity of these barriers. And we have evidence from our own studies. Emily Knox was a graduate student from Boston who's now back at UMass doing a postdoc. And Emily showed that the integrity of the cerebrospinal fluid barrier was out of kilter in these germ-free animals. And she went on to show that we could rescue the effects of various insults on barriers by giving these microbial metabolites, again giving us good evidence how the microbiome mechanistically could be shaping barrier function. Really important, because as our brain ages, if we can really build through nutrition ways to target and produce these metabolites in specific ways, we could actually potentially keep our brain barriers healthier for longer over the lifespan. Another key part of aging brain and nutrition in general is circadian rhythms and sleep. And Gabriel Tofani, who's now at the Monell Institute at UPenn, was a graduate student in the lab from Brazil who made this really important discovery, which we published last year, which is that the integration of circadian signals and stress signals, which again, the stress system is very much wired to our circadian system, but how they are integrated is dependent on the microbiome composition. Because it's not well realized that our microbes go through a circadianicity, as shown up here in the top left, that our microbes change across the day, just like all of our hormones do and everything else. So it may not be just what we feed our microbes from nutrition, but when we feed our microbes across nutrition that may be useful for how stress and circadian signals are integrated. And that might have an impact on how stress-related disorders are manifesting as we age overall. And this is really one of these exciting findings that I really want to build on in the years to come, because it opens up new pathways for intervention that could have long-term benefits for overall health and well-being, especially as we age. So, up until now, I've been talking to you about the microbiome, except I haven't. I've only been talking about the bacteriome. And of course, bacteria are important, but there are other components in the microbiome, the archaea, the fungi, the single cellular organisms, but also the viruses. And we have trillions of viruses in our gut. Now, with COVID, viruses have got a bad rap for sure, but you may not realize that you have these bacteriophages, these viruses that are keeping their deterrents. They're keeping your bad bacteria at bay in your gut. And again, this is a project led up by Nate in collaboration with my virome colleague here, Colin Hill. And what we were able to see was that during chronic stress in adult animals, we were able to show that the virome changes. And then what Nate did was he took the virome from animals before they went under stress and kept giving it back to them as they went under stress to keep the bad bacteria at bay. And in doing so, he was able to attenuate the effects of stress on behavior, on brain, and on the immune system. And this is really cool and deserves a lot more attention as we move forward overall. Of course, this audience is very interested in nutrition, and I just wanted to highlight that in psychiatry over the last number of years, there's been a renaissance in the approaches for looking at nutrition to manage, prevent, and potentially maybe treat aspects of mental health disorders. And the whole area of nutritional psychiatry has emerged. A lot of this work coming from Australia, but these are two European frameworks that I've been involved in over the years, and really showcasing what's going on here. And what we feel now with the microbiome gut-brain axis, that it adds a really important part of the nutritional psychiatry armamentarium, if you wish. And that we don't just have a microbiota gut-brain axis, but indeed what we have is a diet microbiome gut-brain axis. And Elizabeth Schneider is a psychologist from Kentucky who's now was a postdoc, but has now joined the faculty here in Cork. And Elizabeth is really excited about this concept of the diet microbiome gut-brain axis. And we have ongoing work really trying to parse this, how the different components of diet, and ongoing studies with colleagues in UCLA on polyphenols. I'll talk about fermented foods as well as fibers to really show how can they impinge on how the microbiome is signaling to the brain and all these pathways. And that the next time you open your larder or fridge, that you will do it through the lens of your gut microbiome overall. And more recently, in a review just out in the last few weeks, we're really going from fork to feelings through our microbiome overall. And we really think that this is an important way. And one of the key, protagonists or culprits are the fermented foods. And we've been really excited about the potential for fermented foods overall. And we've done studies in animals showing that they can have beneficial effects in various animal models related to social behavior and social deficits. And Kirsten Birding was a registered dietitian that came out of Chicago, Urbana-Champaign, and came to our labs, and she really did a good job in challenging me to be able to, if you believe in that the diet is going to be important, we should design a diet that's high in fermented foods and really ramp up fiber as well, because fiber is king for your microbiome. And if we do that, we should be able to dampen down the stress response. And in this small study, it's in 45 individuals, we were able to show a reduction in stress after 1 month's adherence to this diet, and those that stuck to the diet better had a better stress outcome overall. We're now in the process of extending this to 8 weeks, and because this was a diet in combining fermented foods and fiber, we were interested in really looking at the biology of both of these components. And as we move forward, we're beginning to see that fiber might have a different effects on biology than fermented foods. And that's really one of the things in our human studies we're really trying to look at is what are they, where do they converge, and especially at the level of the immune system, and then how can they have beneficial effects on age-related cognitive decline, age-related changes in social behavior, age-related changes in anxiety, etc. overall. And really with fiber, when you do look at cross-sectional studies across the board, that people who have higher fiber in their diet have better mental health outcomes. That is shown time and time again. Yet when people do studies, interventional studies with fiber, the results tend to be relatively disappointing up until now. And this was a systematic review showcasing that. So we really need to understand how diet can, like fiber, can affect the brain through the microbiome or through a moderation effect, and ongoing studies are like that. I just wanted to highlight a recent study that should come out any day now on coffee, because coffee is also a fermented food, and we showed again that habitual coffee changes the microbiome, and that when we put people into with coffee withdrawal, and then reintroduce coffee or reintroduce decaffeinated coffee, so we could look at different components. We could have beneficial effects on behavior, as well as changes on immune system and on the microbiome. And we think that some aspects of how coffee is interacting with the microbiome could be beneficial for some of the stress relieving and cognitive effects of coffee in these healthy volunteers. That's what our data would show. And so stay tuned for that to come out overall. So in conclusion, really what I wanted to highlight was that the diet microbiome gut-brain axis is emerging now for prime time. We need to build causality and biology into it, but it's not just for psychiatry. It's also going to be relevant for neurology across a variety of disorders, from Parkinson's disease, Alzheimer's disease, and even in epilepsy, where people are already using ketogenic diet for almost 100 years now. So stay tuned to see where this field will emerge. There are challenges. Going from these wonderful data in animals to humans and back again is challenging. And this field is very much about correlation. And you can correlate almost anything. And I had the pleasure of working with Ute Neutling's group in Bonn some years ago, where we could correlate the microbiome composition on cognitive performance in young adults. But you can correlate anything. So you can correlate the divorce rate in the state of Maine with the per capita consumption of margarine. Now, being Irish, we like butter. And so if your partner came home with margarine all the time instead of butter, that could lead to arguments. And those arguments could eventually, if they keep happening, lead to divorce. And these are the kind of stories we're telling ourselves in this field time and time again. So we really need to move towards causality in this field, understanding the effects of lifestyle and other environmental factors, trying to get to grips with what a normal and healthy microbiome is. But that will be that's where large-scale translational longitudinal studies are going to play a key role, and understanding gene-environment interaction. But the good news here is that unlike our genome, which really we can't do a lot about except blame our parents and our grandparents, our microbiome is potentially modifiable. And that gives individuals and patients a potential agency over their own health and well-being, and especially their brain health as they age overall, because through the power of nutrition, we can actually intervene. And so as we move forward, we're beginning to look at this much more. And it's really a team sport involving many different disciplines, ranging from nutrition to psychology to sociology, using big data, much more back translating. And then it's also one of the things that I would want to finish on is also the importance of looking at regional differences. And so one of my graduate students, Ben Valderrama, is from Chile, and he has been challenging us not to have just a global north view on the world, and really we should be looking more across different cultures. And more recently we've looked at the microbiome in neglected populations, especially in South America in particular. And we think also for microbiome gut-brain axis and mental health in general, we need to also not forget about the global south and the importance, and also not just from a charitable point of view or solidarity, but also in terms of what it can tell us about holistic foods and diets and nutritions and culturally sensitive approaches that might open new ways to unlock secrets of how diet, gut, brain interactions occur. So thank you very much. Yesterday was International Women's Day. I got to give thanks to the amazing women on here on the left that I have the privilege of working with, that challenge me and excite me and help us solve all of these wonderful studies around how the microbiome is shaping brain and behavior, and not forgetting the men on the right either, who are to do their share. But I did want to mention that. And I leave you again with the words of Hippocrates, who's thought to have said, and again, it could be fake news, but he's thought to have said let food be thy medicine. I'll change that to let food for your microbes be medicine for your mind. And then just remind you that every now and then, just every now and then, it's good to turn things upside down and look at them in a different way. So I hope I've challenged you to do that today. Thank you very much.
Thank you so much, Dr. Cryan. What a wonderful tour de force seminar. And I really like your concept of work to feelings, which in many cases we have been trained as scientists to not think about feelings and emotions, and to incorporate this makes it really more translational science. So thank you
Thank you,
for
Jen.
that. Just a reminder to the audience to please input your questions into the Q&A chat box. And I think I will start with a question, if I may.
Sure.
I have a few, but I will pace it out. So in terms of, it's so interesting, your feeding interventions with pre and probiotics combined. Did you see that? That seems to be a synergistic improvement, because there are many studies that look at fiber
Yeah.
alone and the result is not as compelling as you may predict. So pairing that with your looking ahead to your future studies, your other story where you're looking at frequency or chronicity of feeding with circadian rhythms. Have you looked to see if there are different times of eating
Oh,
or
it's
patterns of eating, like intermittent fasting?
Yeah, no, we haven't done it, but it's in my next grant for sure, and it's something I get asked a lot about. And really, like where our data now is that we have different pillars. So the microbiome is talking to the brain. There are different pillars of where these interactions are occurring, and circadian is definitely key to that. And that's something that I really, yeah, I'm very strong to think. We haven't looked at time restricted eating. We want to do it. We're interested in fasting in general anyway because of some of the beneficial effects that we know are happening in relation to cardiometabolic and immune health. There's less shown on brain health generally, but I think it definitely is something that we should be exploring more and more. I might stop sharing. Will I stop sharing? Would it be better?
It's up to you. I'll transition to the questions as they're coming in through the chat. So,
Okay.
first question from Brett Rust. If we think of short chain fatty acids as mediating some aspects of the gut-brain axis, what do you think is the most likely conduit? We have found it's difficult through diet to alter short chain fatty acid concentrations in peripheral blood. What is the likely avenue to affect behavior? Vagal, hormonal, directly, or something else?
Yeah, it's a great question. Short chain fatty acids are just tricky to work with no matter what. And what we met because they're so volatile, what we're able to measure in blood may not be, we may be missing some of the tricks. I don't know. I'm not an analytical chemist, but I think there is parts there for sure. Secondly, I think short chain fatty acids and other medium chain fatty acids and other fatty acids interacting with vagal is going to be really important as well. And then also short chain fatty acids at the level of the blood-brain barrier is going to be really important overall. So I think it's a combination of all of that. I think we need to do proper carbon 13 studies and see can we get the short chain fatty acids to what level are they getting to the across, and then what short chain fatty acids, because they're not all going to be the same. Butyrate's going to be different acetate, and but it's a great question. It's a really important question for the field to grapple with overall.
Absolutely. And I guess to follow up, there was a lot of recognition of the blood-brain barrier. Have you, in your studies, looked at the gut barrier directly?
Yeah, I know. We start there, and because it's more studied, and the machinery, there's overlapping machinery in terms of tight junction proteins and various things there. So we've been showing that, like in one of our studies in early life, we were able to show that nutrition was able to help the integrity of the gut barrier overall, and specific strains of bacteria can have beneficial effects in relation to that.
Great, thank you. Second question from Shandong Wang. Thank you so much for a wonderful and comprehensive presentation. I was wondering if the regulation of the gut microbiome by coffee drinking is due to its direct effect on the gut bacteria or its effect on the brain nervous system, which then alters the gut microbiome.
Oh, that's an intriguing question. I hadn't really thought about that, but it does raise another argument. So one of the reasons we looked at decaffeinated was to kind of take out so we did caffeinated and decaffeinated. It's a small study. We did 62 people, was to be able to kind of differentiate the stimulant effects of caffeine. So that helps us. And so there were some effects that were shared between decaffeinated and caffeinated. And also there were some effects in the microbiome that were unique to the decaffeinated as well. But the decaffeinated contains all the fermented and the polyphenols and various things. So there's some direct effects there. But the point that's raised here is really that Zhangdong raises is also about top-down regulation as well as bottom-up. And I think that's really cool because there's really nice studies now that you can activate parts of the brain and change the microbiome to change how it signals back up to the brain. So that's really cool. And these studies are ongoing in animals, but also with neuromodulation techniques in humans, we can do more there as well.
Great. As a follow-up question, I had one, going back to your consistent with Jean Dong's question and to your social brain studies before. Do you think in certain aspects, is it really a top-down or a bottom-up regulation?
Yeah, like the top down has been forgotten about a little bit, but I think we definitely need to explore it more, and I think it's getting more attention.
Okay.
And I have a article under review now where we're trying to do top down, bottom up, just to get people to think about that because we spend so long trying to get from the microbes to the brain. How is this happening? Does it happen? I spend most of my life trying to convince neuroscientists that there are any inputs from the body to the brain, which I think is changing. But I think slowly we're getting there.
Yes, absolutely. Okay, so Dr. Chris Wiley has a third question. Wonderful seminar. I was intrigued by some of the sex specificities in your data. Do you have any insight on how sex interacts with the microbiome to drive these differential effects?
So big question, important question. Where the field is right now is looking at the specific bacteria consortia that are able to metabolize sex hormones. So in females there's an estrobolome and there's also a male equivalent like an androbiolome or testosterone that breaks down and there's lots of really exciting work coming out to show that this is functionally relevant and is able to help recirculate. This will be important during puberty in particular, but also during perimenopause. And we think that's something that people are beginning to get excited about. And indeed, one of my colleagues here is developing a kind of a product of tailored microbial nutrition for perimenopause to try and deal with some of these sex differences. But it also brings up other issues about vulnerabilities that occur in sex differences in brain health that we ignore much to our own detriment overall. And the male and female brain are quite different, and why wouldn't they respond differently? I guess is part of it.
And we have another question from Richard Alpert. Thank you for such a wonderful, energetic talk. You have given us so much food for thought. Now, please excuse the banality of my question, but has your research produced any changes in your own personal behavior?
Oh, that's a good one. Well, I don't know about my behavior. Well, I have to ask my wife that. But in relation to my own personal practices, perhaps more than my behavior, is two things. One is I'm more conscious of fiber. You're more conscious of what fiber you're taking in, and you're trying to look at that and build that into your choices. And secondly, in relation to fermented foods, I try and have milk kefir and or kombucha as much as I can. I travel a lot and they die in my. I'm not a good. My poor kefir grain hates me because But anyway, I keep going back to the same source and rejuvenate. But I'm more conscious of that. And if I'm giving people something practical, and the great thing about fermented foods and fiber is that they don't have to cost a lot. We can democratize brain health,
Right.
and we can get to lower socioeconomic areas which might benefit from this overall. But in Ireland, we have a really poor culture of fermented foods. We don't ferment overall. The best we have is some yogurts and cheese, but other than that, we're not good. So trying to build that into is a culturally important aspect.
Well, thank you so much for your time. We're nearing the top of the hour and want to be mindful of your 5 o'clock meeting coming
No
up.
problem. No problem.
So, to the audience, thank you so much for joining today's seminar series and for the great questions. And to Dr. Cryan, thank you for just contributing such important science for the many decades of your research program. And we look forward to seeing all the new publications with these exciting works in coming years.
Oh, thank you, Jen, and thank you so much for the invitation. It's been a real pleasure. Hopefully, I see you in Boston at some point.
Please let us know. Thank you so
Yeah,
much. We'd love to have you in person.
no problem.
Bye.
Bye, bye, bye.